Inside every single one of your cells is the ultimate blueprint for life, your DNA. It's the master code for everything that makes you, well, you. But for life to go on, for your cells to divide and make new ones, this entire intricate blueprint has to get copied. And it has to be perfect. So let's dive into one of biology's greatest heists.
Think about that for a second. How does your body pull that off? We're talking about a code that's got 3 billion letters. It's an insane amount of information. And it gets copied with near-perfect accuracy, trillions of times throughout your life. This is way more than a simple copy and paste job. It's one of the most incredible, coordinated operations in the entire universe.
So why bother? Why go through all this trouble? Well, the mission is simple, but it's absolutely critical. It's all about survival and continuing the story. Every time a cell divides, the new daughter cell needs a complete, flawless instruction manual to know how to function. Without that perfect copy, things can go wrong very, very fast. Okay, so let's get the mission briefing straight. The why is to give that new cell a full set of instructions. The when is during a special prep stage called interphase, which happens right before the main event of cell division. And the where, for this whole high-stakes operation in cells like ours, is inside the most secure location in the cell, the nucleus.
Now, you can't pull off a mission of this scale with just one molecule. No way. You need a specialist team, an elite crew of enzymes, where every single member has a critical job to do. The whole thing depends on flawless teamwork and coordination. So, let's meet the main players on this molecular heist crew.
First up, we've got helicase. This is the unzipper. Its whole job is to get in there and pry apart that famous double helix. It literally just plows down the molecule, breaking the weak bonds holding the two strands together, which opens up the code so the rest of the team can actually read it.
And that brings us to the star of the show, DNA polymerase. This is our master builder. It follows along the open strand and just starts building the new matching half. It knows the rules by heart, meticulously adding the right partners, A with T and G with C. It's incredibly fast, and, as we're about to see, it's unbelievably accurate. But here's the catch with our master builder, DNA polymerase. It's a genius, but it can't start working on a blank slate. It needs a signal, a starting block. And that's where primase comes in. Primase is the initializer. It lays down a little piece of RNA called a primer, which basically acts like a giant neon sign that says, hey, start building right here.
And finally, we have ligase. This is the gluer, the finisher. As you'll see in a minute, the building process isn't always a smooth, continuous line. Sometimes it has to happen in little chunks. Ligase is the one that comes in at the very end to seal all the gaps and make sure the new DNA is one solid, unbroken strand.
All right, the team is assembled. Now, let's see them in action. This whole operation doesn't just begin at a random spot. It kicks off at very specific locations on the DNA called origins. You can think of these as the designated entry points, the official starting gates for our molecular crew.
So, the mission starts. Once the origin is found, helicase gets to work unzipping. But you know, those single strands are kind of sticky. They really want to snap back together. That's where a support crew comes in. These little SSB proteins act like tiny doorstops, holding the strands apart. Meanwhile, as you unwind something like a rope, it can get super tangled up ahead. That's what topoisomerase prevents. It's our knot prevention expert, relieving all that tension so the whole operation doesn't just seize up.
Okay, so everything seems to be going smoothly. But just as the team really gets going, they hit a major snag. A huge complication that's baked right into the fundamental design of DNA itself, and it makes a simple, straightforward copy impossible.
And here it is. This is the core of the problem. The two strands of DNA are anti-parallel. That means they run in opposite directions, kind of like a two-way street. But our master builder, DNA polymerase, it's strictly a one-way worker. It can only build in one direction, which we call 5' to 3'. So how in the world do you copy both sides of a two-way street when you can only travel in one direction? The cell's solution is nothing short of genius. It uses two completely different strategies at the exact same time. On one strand, the builder can just cruise along, following right behind the helicase as it unzips. It's a smooth, continuous process. We call that the leading strand. But for the other strand, it's got to work backwards, away from the direction of unzipping. And this is the lagging strand, which requires a bit more finesse.
So this lagging strand is built in this incredible little loop-and-build dance. Primase pops on and lays down a starting point. Then DNA polymerase jumps on and builds a small fragment, backwards. These are called Okazaki fragments. Then as more of the DNA unzips, the whole process repeats further down, another primer, another fragment. And finally, our finisher, ligase, zips along and glues all of these separate pieces together into one perfect continuous strand.
So after all that unzipping, building, proofreading, and gluing, the mission is complete. From that one original DNA molecule, the cell has created two absolutely identical copies. Each one is now ready to be passed on to a new daughter cell, a perfect blueprint for life.
Now, scientists have a pretty cool name for this whole process. It's called semi-conservative. It sounds a little complex, but the idea is actually super simple. It just means that each of the new DNA molecules is a hybrid. It's made of one old strand from the original molecule and one brand new strand. The cell cleverly conserves half of the original in each new copy. And the precision here, it's just breathtaking. The error rate for this entire massive operation is less than one mistake for every billion letters it copies. To put that in perspective, that's like typing out all 7 Harry Potter books more than 700 times and making only a single typo in the entire thing. It's just staggering.
So how on earth is that even possible? Well, our master builder, DNA polymerase, has a secret weapon. It's not just a builder, it's also its own quality control inspector. It has a built-in proofreading function. So as it adds a new letter, it literally double-checks its own work. If it catches a mistake, it can actually stop, go back, snip out the wrong letter, and put the right one in. It's just unbelievable.
So why does all this molecular detail really matter to us? Because this process right here, this is the engine of life itself. It's how we grow, how we heal from a cut, how we pass our genes to the next generation, and maybe most importantly, understanding this mission lets us intervene when it goes wrong. In fact, many of our most powerful medicines, from antibiotics to chemotherapy drugs, work by specifically targeting and jamming up this replication machinery in harmful cells like bacteria or cancer.
It really just leaves you in awe, doesn't it? This incredibly complex, perfectly choreographed, and nearly flawless operation is happening inside your body, billions of times right now. And it really makes you wonder, if this tiny molecular team can pull this off, what other incredible secrets and complex codes are still waiting inside our cells, just waiting to be cracked?